Okay, so video for this week. Some of you may be pleased to know that you won't get motion sickness this week because I've invested in a proper camera stand, so it won't look like I filmed the last video on a boat. Okay, so today's topic is enzymes, and hopefully by the end of the video you'll be able to answer these three questions. So enzymes, what are they? How do they work? And what changes how they work? So to address the first of those questions straight away, enzymes, what are they? Enzymes are biological catalysts. So these are the two important words, biological catalysts. And in the context of this sentence, what does the word biological mean? Well, we call them biological catalysts because they're made of proteins. And what is a catalyst? Hopefully you know this from chemistry. So a catalyst is something that speeds up a reaction. So when we say enzymes are biological catalysts, so they're biological, they're made of proteins, and they're catalysts because they speed up a reaction. So straight away, go back to this one. Enzymes, what are they? Biological catalysts. So a little bit more about that. And for that, I'm going to need to bring in my laptop. So this is the height of technology. All right, I'm going to edit that out so it was seamless. But actually, in reality, that was a 5-minute pause where I had a big fight with my laptop. But you'd be pleased to know that I won. So here we go. Right. Can't ever say I was conventional. Filming a laptop horizontally on the floor with a mobile phone. So here we've got 3 different molecules on this side, different molecules over here. And so enzymes, they're biological catalysts. I'm going to say that a lot. They speed up a reaction. And there's 2 types of reactions really that we're interested in. Digestion, which is where stuff gets broken down. So one big, long, complicated molecule gets broken down into smaller molecules. Or synthesis, which is creating stuff. So if something's synthetic, we say it's man-made because it's been created by us. Or if you synthesize an antidote, you've created an antidote to a disease. So if an enzyme does digestion, it breaks a molecule down into smaller molecules. If an enzyme is synthesizing something, it's turning small molecules into big molecules. So looking at this, so what we've got here, zoom in, right. And that wasn't actually a zoom, that was just me moving my camera stand. Super technical now. Right. So we've got bits of a protein molecule here. Protein, massive, huge, long molecule. If we were breaking that down, if we were digesting the protein, proteins are made of amino acids. So you eat some protein, so some fish, some meat, some eggs, gets broken down, and your body breaks it down to amino acids. And then when all the amino acids are in your body, you might want to use those amino acids to build your own protein if you were going to build some more cells, build some more muscles. And so that would be synthesis. So you can turn amino acids into protein using an enzyme, or you can break protein down into amino acids. So this is starch. Starch is an example of a carbohydrate. Starch is found in like pasta or potatoes or things like that. Your body can break down starch into glucose, or it can combine glucose, synthesis this way, back into starch, and then lipids or fats. This one's slightly different because these are all the same, these are all the same. Lipids, it's made of 2 separate things here. If you break down a lipid, a fat molecule, it breaks it down into fatty acids and glycerol. So you've got examples there of how your body might use enzymes to do digestion or synthesis to create things. Right. Now, in terms of how enzymes work, do you know what? I am just going to go back to this. And here, so some specific examples. So you've got amylase, which you find in your saliva or in your small intestine, and that breaks down starch into sugars. So if you have something starchy and you chew it for long enough, keep it in your mouth, get it really moist with loads of saliva, it should start to taste sweet. So the longer you chew it for, the amylase will break down the starch into sugars, and you'll start to taste sweet in your mouth, like a wafer from an ice cream cone or something. If you chew the wafer's just made of starch. The wafer isn't sweet, but if you chew it for long enough, it will start to taste sweet. Catalase, found in lots of cells, but mainly in the liver, breaks down hydrogen peroxide to make water and oxygen. Starch synthase, so synthase linked to synthetic or to synthesis, it's going to be creating stuff. So starch synthase found in a plant. Synthesis, so create starch from glucose. And DNA polymerase, phononucleus, it's how your body creates DNA. So it uses all the individual monomers, so like little small molecules, and creates DNA from them. Right, in terms of how that works, so that was our second big question. So enzymes, we know what they are, we've talked about some examples. How do they work? And the example that you need to know for that is the lock and key hypothesis. As you can tell from my amazing diagram of key and a lock. So, as long as it's a decent lock, locks only fit one specific type of key. You can't use your car key to open your front door. You can't use your front door key to start a car. You can't use your locker key to start a car. Ran out of examples. But the point is, a key is specific to a lock. Each key only fits one lock. And it's the same for an enzyme. So here's one I drew earlier. Lovely, beautiful enzyme here. And the bit we're interested in is this part of an enzyme, which we call the active site. That's the really important part of the lesson, the enzyme. So this part here is the active site. And enzymes work on things called substrates. So substrate floats along. Again, here's one I prepared earlier. Substrate floats along, fits perfectly into the active site, and then the enzyme can speed up a reaction, break it apart into two smaller things if it was digestion, and out it goes. So where's this going with the lock and key? Well, the active site is specific. It doesn't fit other substrates. So as an example, I've got a different substrate here. This substrate does not fit, it doesn't match the shape, it won't fit in that active site. This one doesn't match perfectly the shape of the active site. So this enzyme will only work for this substrate. If we were going to draw an enzyme for this one, let's see how accurate I can be. Probably not very. Right, so that enzyme should fit this active site, should fit that substrate, close enough. Whereas this one, no chance. This one, no chance. So that's an example. That enzyme fits in the substrate. So the substrate fits into the active site of the enzyme, and then the enzyme can react it, break it down, digest it, and off it goes. Or if we were going to do an example of synthesis, let's just cheat quickly to make this a little bit easier to draw. So, draw on the floor. Nobody tell miss. Please roll off. Come on. Right, there we go. So, got my enzyme, got my weird looking active site. Should have drawn this off camera. Right. So this would be an example. This enzyme fits, this substrate fits into there. That substrate fits into there. What have I drawn? Right, there we go. So that will fit in there, that will fit in there, and then these two can join together and float off as one molecule. So they come in separately, get joined together, and off they go. So lock and key means the active site of an enzyme is specific to one substrate. So back to our big questions. What are enzymes? What are they? They're biological catalysts that speed up a reaction. Biological because they're proteins. How do they work? Well, lock and key hypothesis. So the substrate fits into the active site, and then if it's a digestion reaction, it will break the substrate into two small substrates. Or if it's a synthesis reaction, two substrates will go in, join together, and leave. And we've talked about some examples like amylase. And last part, what changes how they work? Well, there's three things. And first one, we'll talk about concentration of substrate. And I'm going to draw a graph, concentration of substrate, and this is going to be rate of reaction. The graph looks like this. And just to explain that, so what's actually happening with this graph? As you increase the concentration of substrate, the rate of reaction increases. Rate's a posh fancy word for speed. So the more substrate there is, the faster the rate is. But when you get over here, we're having more and more and more and more and more and more substrate, higher concentration of substrate, and it's not affecting the rate of reaction at all. And so for that, I'm just going to talk about an example. If I was going to do a bird's eye view of foyer lobby of a cinema, so got the counter here. Let's have a couple of tills, 3 tills. Let's have head and shoulders of employees. Right, and then we'll stick a customer in there. So these are our employees, these are our 3 tills, we got 1 customer. How fast can these guys sell popcorn? At the moment, not very fast, because we've only got 1 customer. So a customer queues up, buys it, we've got these employees doing nothing. If we add more customers, so we'll give this person a customer to serve, and we'll give this person a customer to serve. So we've added more customers, these guys can sell popcorn quicker. But if we add more customers, and I'm not going to draw them all, but if I had like 50 billion customers, so the entire population of 8 planet Earths crowds into the foyer of the Odeon, it's not going to affect how fast these guys can serve, because there's only 3 tills, there's only 3 people. So at the start, adding a couple of more customers sped up how quick they could work. But at the end, if we fit the entire population of the universe in here, these can't go any quicker because they're working flat out as fast as they can. How does that link to this? Well, if we increase the concentration of substrate, it means the enzymes can work quicker. The enzymes are going to bump into more substrate molecules. That's going to go click into the enzyme, float off. If there's loads and loads of bits, loads and loads of this substrate, they're going to work quicker. But when you get to this point, it means there's so much of the substrates, the enzymes are working as fast as they can. So in our cinema example, the employees are like the active site. They're selling the popcorn, they're like the active site of the enzyme, and our customers were like the substrate. So if we have more customers, we can sell stuff quicker, but then you get to a point where here, all the enzymes, all the employees at the cinema are working as fast as they can, and so adding more customers, more substrate, doesn't affect it. They can't go any faster. So the second thing, I'm going to keep rate of reaction on there. The second thing that can affect the speed of the rate of a reaction is the pH, so how acidic something is. And this graph looks like that. Line it up so it's reasonably square. So here, if the pH is too low, the rate of reaction is really, really slow. If the pH is too high, the rate of reaction is really, really slow. But if you get the pH just right, like Goldilocks, so if you get the pH just right, the rate of reaction increases a lot. So what we call this point here is called the optimum pH. Not the maximum, not the minimum, the optimum pH. And at the optimum pH, the rate of reaction is maximum. Now this changes for different enzymes. As an example, if you've got an enzyme in your body called pepsin. Pepsin works in your stomach. Your stomach is acidic. The optimum pH for pepsin is 1 or 2. It works really nicely in acidic environments. So for pepsin, it would look like that, because it works really well at pH 1 or 2. But for amylase, which works nicely in your saliva, in your mouth, that doesn't work very well in acidic conditions. That works better at pH 7, more neutral conditions. So the optimum pH can be different for different enzymes, but every graph for pH will look like this. What's happening down here? pH is too low. It affects the shape of the active site. And if you change the shape slightly, the substrate can't fit in so well. So pH is too low, the shape of the active site changes a little bit, substrate won't fit in as well. pH too high, exactly the same thing happens. optimum pH, perfect, quickest rate of reaction. And then the very last thing, and this one is slightly more complicated, but not too much, is the temperature. So for the temperature, it's a bit of a combination of the previous graphs. The temperature graph looks like this. And if we talk about what's happening, so down here, the temperature is so low, and if you think about back to chemistry in terms of what happens to atoms and molecules when the temperature gets really low, the colder something is, the slower the molecules are moving. So, just looking for a blank sheet. So you've got your enzyme with your active site, you've got your substrates, but the temperature is so low that they're barely moving and they don't bump into each other very often. If you increase the temperature, you increase the rate of reaction. And that's because these things start vibrating and moving around quicker, and they're moving faster and faster and faster, and the faster they move, the more they bump into each other, the faster they react. So you might wonder, why does it get low? If what I've said is true, the higher the temperature, the faster they move. The faster they move, the more they're colliding, and that should mean they react quicker. And the problem is, when you get up here, they are moving a lot quicker. They do collide more, but they can't work because the active site of the enzyme changes shape, which is a real problem. So if you imagine, like if we talk about a lock and a key, so you've got your key, it's a really hot day, you get into your front door, and the heat has changed the shape of your lock slightly, and it might mean it's a little bit difficult to fit your key in. You can unlock it eventually, but it takes a while jiggling the key in the lock before you can get it to unlock. And so when you've increased the temperature up here, what's happened is you've started to change the shape of the active site, and it makes it, even though the substrates have got more energy, they're colliding more often, it doesn't fit into the active site as well. And so the rate starts to suffer a little bit. So the rate slows down. And then when you get down to here, this is sort of cataclysmic. What happens here when it doesn't fit at all? The enzyme, we say it's been denatured. If you imagine going back to the lock and key, if I heat the lock up, it makes it difficult to fit the key. But if I cool the lock back down, the key would still fit in. But if you get to a point, if I heat the lock up hot enough that your lock melts and becomes like a puddle of molten metal on the floor, if you cooled that back down, all right, it turns back to a solid metal, but it doesn't turn back to the shape it was. So if something's denatured, it means it's permanently changed shape. Lots of people think it's denatured here. They think as soon as it changed shape, that's it, it's denatured. Denatured is when it's permanently changed shape and it can't go back. Here, it's changed shape enough that it's slowing it down, but if you change the temperature back down, it would be fine. Just the same as the pH, this temperature where it works quickest is optimum temperature. And just as an aside, so your body temperature, 37 degrees. Most of the enzymes in your body have an optimum temperature of 37 degrees, which is handy. In terms of washing powders, if you're washing your clothes at home, you get biological washing powders. They have got enzymes in them, and those enzymes break down fat and grease and dirt on your clothes to help make your clothes clean again. And that's why if you're washing with biological washing powder, you should wash your clothes at 30 or 40 degrees because the enzymes will work best. If you use biological washing powder and put the temperature of the washing machine to 80 or 90 degrees, you're going to denature the enzymes and it's not going to work properly. So going back, last bit of summary. Enzymes, what are they? Biological catalysts. They're proteins that speed up reaction. How do they work? They've got an active site. The substrate fits in. They can either break stuff apart, like digestion, or join things together, like synthesis. And what changes how they work? The concentration of the substrate. The more substrates, the faster they work until it plateaus, like think the cinema workers selling popcorn. The pH, Low pH don't work, high pH don't work, perfect pH is the optimum. And then the temperature, low temperature, the molecules aren't moving fast enough. High temperature, start to speed up, and then you get the optimum, and then above the optimum, even though they're moving faster because they're at higher temperature, the graph crashes down because the active site changes shape. Hopefully that will make sense to you. Have an average week.